I am delighted to welcome Evan Eichler as our first speaker for this season. Evan is a professor of genomic sciences at the University of Washington. He's been a Howard Hughes Institute member for probably about 10 years or so now. His undergraduate degree was at the University of Saskatchewan, which is interesting. Then he went off to Baylor, more standard genetics track off to Baylor. And then I think he was at Case until 2004 or so, at which point he went off to the university to move to Seattle. Evan is extremely well-known and renowned for all of his work in structural variation in the human genome. And the Howard Hughes website says that no major project can be conceived without having Evan involved in it. He's won numerous awards, including from ASHAG, the Kurt Stern, the Newcomb Prize from AAAS, selected to the National Academy in 2013, and has trained many postdocs and students who've gone on to illustrious careers, most notably, of course, our own Andy Sharp, who unfortunately is not here. With that, I will stop and let Evan give his talk. Afterwards, as you know, we will we have mics set up, and we'll have a relatively extensive question period if everyone will stay. Thanks. Bye.
So thanks, Pam. It's a pleasure to be here and give this lecture. I feel a little bit this is Coles to Newcastle in a way. I'm going to be talking about the work that we have done, but a lot of the things that we have done with respect to autism is work that's also been paralleled here at Mount Sinai and other places over the last, I would say, 4 or 5 years. So I'm going to be talking about autism spectrum disorder, and my perspective is that it is a type of neurodevelopmental disorder. One can argue with that, but it's typically or historically was characterized by this triad of impairments: social, behavioral deficits, repetitive behavioral problems, and speech developmental delay. And although cognitive deficits aren't part of the diagnosis of autism, we know, most of us know, that in fact a significant fraction, usually more than 50% of the children that are diagnosed with autism also have neurocognitive dysfunction. CDC places the estimates of the disease now at 1 out of 68. Autism spectrum is, however, an umbrella disorder, and I would argue that that umbrella has actually gotten wider and bigger over the years. And there's obviously significant comorbidity with other diseases such as epilepsy, other neuropsychiatric disorders, including schizophrenia, as well as ID. Now, for the genetics of autism, it's been recognized for over 30, 40 years that there is a strong genetic component. And this has come primarily from identical twin studies, as well as looking at in the context of families, where 70 to 95% of identical twins and somewhere between 5 to 20% of fraternal twins have an increased risk of developing the disease compared to the background, about less than 1%. The other thing I want to talk a little bit more today about is the gender bias. It's not uncommon for most neurocognitive neuropsychiatric diseases to know that males are more likely to be afflicted, but in the case of autism, it is particularly pronounced. So by the old DSM-4 criteria, it's 4 males to every female. And at the time when we still referred to Asperger's as opposed to high-functioning autism, the numbers could be as high as 6 to 8 males for every female that is affected. So I'm going to be talking a little bit about what I think we're learning from genomes, starting from copy number variants to exomes regarding this bias. I would say from 2001 to 2005, there was a lot of effort put into in terms of looking for genome-wide association signals because the disease is so common. We could have been lucky and we could have may have found a few loci that might explain a significant contribution or significant proportion of the risk. Relatively few regions withstood replication, and relatively few loci emerged from the GWAS studies. We knew from work in Mendelian genetics for years before that there were rare inherited forms that were transmitting within families, but the majority of families are simplex. Estimates of 75% of autism families, there's no family history of at least autism in those. So the question is, where's the rest? And I would argue that work done from our group, the days of Andy Sharp, Mike Wigler with Jonathan Sabat, and some of the work from my European colleagues looking at children with developmental disability, Bert de Vries, really helped shed light in terms of the importance of large-scale copy number variation with respect to autism, as well as neurocognitive diseases more general. So it's now, I think, well established that there's an excess of large deletions and duplications associated with autism. Some of the early studies suggest as high as 10 to 1 in families with no family history or simplex autism, compared to what you'd expect in controls or unaffected siblings within those families. In our own work, we started by a completely different approach. We didn't come at it from looking at autism. We started by looking at children with neurodevelopmental disabilities, kids that had gone through standard genetic testing in the 2000 and had come back negative. And the way we looked at it was to look at the genome, identify regions of the genome that were predisposed to recurrently microdelete and microduplicate, and target those regions specifically. So we started with Andy Sharp, some of the first microarrays that were used to target the hotspots of the genome that were prone to microdelete and microduplicate. And this was one of Andy's first papers in 2006 in my lab, which was a recurrent microdeletion, now called the Coulin-de Vries syndrome. We discovered it at the same time as Bert de Vries, as well as Charles Shaw Smith from Nigel Carter's group. A recurrent microdeletion where all deletions, and I think this is the case to this day, all deletions are sporadic. That is to say, this deletion is never inherited or transmitted from a parent, and the children have syndromic features, and you can see this here. They're all intellectually disabled. They have this kind of characteristic facial features, this bulbous nose, protruding tongue, and this is now a well-recognized syndrome. It turns out it's almost exclusive to individuals of European and Mediterranean descent, and I'm happy to talk about that if anybody's interested in terms of why that's the case. Shortly thereafter, Andy found a second microdeletion. So this is just based on screening children with unexplained developmental disabilities. This turned out to be an autism spectrum disorder. All cases that have been identified were identified in 2007. We had 6 initial families. They all had a recurrent microdeletion of about 3 megabases located on this region of chromosome 15q24. So in addition to a diagnosis of autism, when we got to 3 or 4 families, you could see the syndromic features. You can see, for example, this frontal bossing of the forehead, this almond-shaped eyes. What you can't see from this picture is that these kids are in the 25th centile of growth. So these are children that are particularly small and also have autism. This is the last example I'll show you for a copy number variant, discovered also by Andy. So this is the Andy Sharp slide from my lab. This is actually not strictly just de novo. So this one was interesting because this is one that we discovered actually early on. So in our first few hundred children that we screened, we found this deletion. And the reason that we didn't follow it up immediately is because we found it transmitted. And it was transmitted in 2 of the 6 families that we found. It was transmitted from one of the parents. And so we thought, well, that's not as important or as highly penetrant. So we focused on others. And then subsequently, we started screening, instead of a couple hundred, we screened thousands. And then we started screening tens of thousands of normal controls. We never found this deletion in the normal control population, or almost never. So we knew it was a significant risk factor, and when we went back and tracked the families, we could see that half the time it was inherited, half the time it was de novo or sporadic. And in our families that we studied, there was no obvious dysmorphology associated with these individuals, but they were all with borderline intellectual disability, but this emphasis here was on borderline. So we reported this in 2008 as a recurrent microdeletion just distal to Prader-Willi on chromosome 15q13. And then shortly thereafter, I think a paper came within months from Jim Lepsky describing the same. And now that we've screened on the order of 30,000 patients, and we now have hundreds of families with this microdeletion compared to controls, which is almost non-existent, we know that the odds ratio of this particular variant is very high in terms of actually being a risk factor. But it's probably not necessary and sufficient, at least for severe developmental disability. And in work that came just, I think, really within 6 months, it was one of the first CNVs, I think, associated strongly with schizophrenia. So in work that we followed up with looking at families that have epilepsy, we now know that this particular CNV is found in 1% of individuals with idiopathic generalized epilepsy versus 0 out of matched controls from the same types of populations. So this is a nice example of where life gets complicated in genetics, where we are confident that we have a strong risk factor for, in this case, autism and intellectual disability and epilepsy, but it's probably not necessary and sufficient to always give you the phenotype. So keep that in mind as we start to look at genomes or in exomes and genomes. So that was our initial foray. We started with looking at hotspot regions, identified about 12, and I think now it's on the order of about 20 recurrent microdeletions of what is now 50 of these. Most of these are mediated by segmental duplications in the human genome. And in my lab, a number of individuals started to look at this question more broadly, looking at large-scale copy number variation, irrespective of whether it mapped to these hotspots or across the genome. And so this is essentially analysis, which is now out of date, but this time we looked at about 8,000 controls versus 15,000 patients. These patients all came into the clinic with developmental disability. The 8,000 were actually controls from GWAS studies where we recalled the actual CNVs in those. And this line is essentially the burden of CNVs in controls in black versus cases indicated here in red. And so for any given position, for example, at 500 kilobases, you can see that about 25% of kids that came into a clinic with some form of developmental disability have a large CNV, 500 kilobases or greater, compared to about 10% of you guys sitting in this room. At least that's how I would interpret it. And this and work that was done by David Ledbetter around the same time led us to suggest that, at least in terms of a significant high impact risk factor, large copy number variants defined here as greater than 500 kilobases account for about 14.2% of all developmental disability. Now, if you think about that, that's not chump change, right? The estimates are that 3 to 5% of births, live births, have a developmental disability. And in this case, 15% of it is due to this type of variation or this form of large-scale deletion or large-scale duplication events. Not every disease shows this effect. So this is a number of different pediatric diseases that we've looked at for CNV burden. Exact same plot that I showed you before. The green line is the control line. And here we're looking at kids with craniofacial, and this should be really congenital heart defects as opposed to cardiovascular, epilepsy, autism, and asthma. And you clearly see that there are diseases, for example, like craniofacial defects or congenital heart defects, where the actual burden of CNVs is in fact higher. So these lines are far above what we would see in epilepsy and asthma. So the estimates now, some work from Matt State, suggest that the actual contribution of large CNVs, here defined typically at 4 to 500 kilobases, is on the order of 7 to 8% for such diseases. And then for diseases like craniofacial and congenital heart defects, it's higher. It's closer to be double, past double that effect. And then you look at other things, such as children with asthma, and there's absolutely no burden for these large CNVs. So this is actually kind of important, because if the large CNVs are contributing their effect primarily through de novo or private mutations, so they're private to the family, this is like the canary in the mine shaft, telling us which diseases we would expect to see de novo types of mutations playing an important role. It doesn't say that there isn't forms of asthma that are caused by CNVs. It just says that there's not enough in a general population screen to see this burden. Last slide I'll show you from the CNV world is this one. This one caught our interest. We started seeing these effects back in 2010, and it really became significant in 2014 when we really had larger numbers. So these are families that have come in for developmental disabilities which had a CNV. And what I'm indicating here is the proportion of events which are either inherited or de novo as a function of size. Blue means they're inherited from father. Red means they're inherited from mother. And I've broken out the deletions and duplications these are all clinically ascertained families, but where we actually know the mother and the father CNV pattern for all of them. So 2 important points. Compare deletions versus duplications. You can see, pretty much no matter what size threshold you use, that the proportion of CNVs that are de novo for deletions is much higher than duplications. And this is not to be unexpected. It's been known in model organisms like Drosophila for years that deletions are much more deleterious than duplications. So the way they're going to arise in a population is primarily by de novo mutation as opposed to essentially inheritance, especially when they get big, because their selective effect or effect on fitness is in fact much greater. But this is the part, if you have a really good eye and you can actually integrate across the area, you will see something here that for a geneticist is screaming red flag. If you look at what is transmitted, what you find that in fact there's a significant difference in the proportion that's transmitted from mother or father. So it turns out for event sizes greater than 400 kilobases, one marker, 60% or 58% of the events in these 2,000 families were coming from the mother as opposed to the father. This is not Mendelian. It should be transmitted equally from either side, unless there's some type of gender bias with respect to the transmission itself or carrier status within a family. So this was the first inkling that we had, that in fact, females are more likely to be carriers of these types of potentially deleterious mutations. And so you can see the effect. In fact, it even looks like it gets a little bit stronger as the sizes get larger. So I want you to keep this in mind, because I'm going to come back to this with exomes and then also with the genomes. So I could go on forever about large-scale copy number variations and the things that we've pursued with them, but I'm going to stop there. And I'm going to tell you what I think is important about the lessons that we learned from the large CNV screens that were done on kids with developmental disabilities, epilepsy, and autism. The first was that gene dosage and balance is really important in terms of the etiology of disease. Because in almost all the cases that we found, the events were heterozygous. So we're not seeing homozygosity here of a specific lesion. We're seeing a deletion or a duplication of typically a dozen genes associated with these large events. In those that were actually the most clear syndromic, and therefore the most highly penetrant, they were almost always sporadic mutations. So heterozygous gene-killing mutations, i.e. taking one copy as opposed to both copies of the gene out. Other important message, individually rare, because each one of those sharp syndromes was 0.3%, 0.2%, 0.1% of all cases, but collectively common. So altogether, the estimate is on the order of 14 to 15% of cases with developmental disabilities, 7 to 8% of cases with the DSM-4 diagnosis. Third, when we see transmission, mothers are more likely to be carriers, suggesting that there's, in fact, perhaps a different buffering capacity for these types of mutations depending upon your sex, male or female. So CNVs are great, and they continue to be great, and I think they're going to be the wave of the future as we move forward into really rigorous genome sequencing, because we'll be able to actually survey this landscape below 50 kilobases, which really has been opaque to us over the last few years. But there are two main problems with CNVs. First, most of the CNVs that we know now are pathogenic or important risk factors are large. So they affect dozens of genes. And so if you're interested in the gene and the pathway, good luck, because many of these CNVs, we've never been able to reduce them down to a specific gene. It's more than half the cases, it seems like we never see a reduction of smaller deletions to pinpoint a specific gene. The second is that even though we're doing the screen, we have now screened over 50,000 cases for large CNVs as well as controls, in many cases we're still finding one-offs. We're finding a CNV that we've never seen before. And so a very large fraction of CNVs, although we can predict in aggregate the statistic that they're important, for that specific family, we can't say it's the cause to be absolutely certain of the disease in those cases. So large CNVs suck precisely because they're extremely rare and they're large with respect to this. So at least the large thing could be addressed in principle if we could routinely sequence exomes and genomes, and the same principles that we learned in terms of gene-killing mutations essentially was a perfect application for the advance of next-generation sequencing. So when this technology came out in 2007, it really became mainstream in, I would say, about 2009, 2010. The idea that we could go back to the very same families that didn't have the large CNVs and discover now and test the model of disruptive mutation events that were de novo or private as association with disease essentially was an immediate application of this. And the cost made this essentially doable to do not just hundreds, but ultimately thousands and now tens of thousands of families. So we applied exome sequencing. So we started developing and using it in our lab. First publications in 2011 in autism families, beginning to look for kind of the same type of model. Instead of focusing on the whole genome, focus just on the protein coding bits. And let's look specifically at families in which they are simplex and they have a diagnosis of autism. So we had the good fortune of having access to the 2,500 families from the Simon Simplex collection, which were collected precisely from the perspective that mother, father, an affected child, and one unaffected sibling had been collected with deep phenotyping information. There was no family history of the disease, and therefore there would be a potential enrichment for de novo mutation events. And the idea was very straightforward. For all those initially that didn't have large CNVs, let's go back in and let's resequence these families And let's look specifically for the pattern of de novo mutations and compare the pattern that we see in the affected proband versus the unaffected. So almost like a natural genetic control within the family. So focusing just on de novo events. And the question is, do we see an excess of gene-killing mutations in the proband compared to the unaffected sibling? So I'm not going to bore you with all the papers that came out in the first couple of years because you've heard enough about them, but I'm going to summarize all the studies with essentially this slide. So I would argue that 2012 was a banner year for exome sequencing of autism families. There were 4 studies that were published, one that you guys were involved in, one that was from our lab, from Brian O'Rourke, where all in total about 650 families were sequenced. About half of them were of this design where we had an affected and an unaffected sibling. And the results for ASD were really exciting. The good news, and this was seen and now has been seen over and over again, when you compared the affected versus the unaffected, there was no difference in the actual mutation rate. There was almost no difference between the mutation rate between the two. But when you looked at the type of mutation, you saw a 2-fold X of de novo gene disruptive or putative gene disruptive mutations like splice sites, frame shifts, gene disruptive events in the affected compared to the unaffected. And a 2-fold signal based on the first 650 families was tremendous. So in other words, a child with autism is twice as likely to have a de novo mutation event that kills the gene compared to the unaffected sibling in the context of the same family. I think anyone working in the field of autism was tickled pink with this observation, because that meant that the CNV observation of large deletions being found in excess in probands was now also being seen at specific mutations within genes as opposed to these large events. There was bad news, and the bad news was this. So everybody, well, not everybody, actually we shared our data sets pretty early on before the papers actually even came out, but we were kind of holding our genes because we thought, well, maybe we would actually find the autism gene. And maybe when we didn't see recurrences, so the same gene hit twice very often, we thought, well, when we compare it against, Ben Neal's data set and Matt State's data, it's all going to become clear. And the answer was no. We almost never saw lightning strike twice. So there are only 18 genes where a de novo mutation occurred in the same gene. And if you just limited it to likely gene disruptive events, there were only 5 genes where we saw a recurrent likely gene disruptive mutation event. So this was sobering, because it meant that everybody's collections of autism patients, and some in the numbers of thousands, was underpowered to prove the pathogenicity of any given gene, just simply. So we couldn't say for sure, even the genes that had 2 recurrence, although we were probably 95% certain that they were a risk factor, we couldn't be absolutely certain with respect to this, with respect to the families. So fast forward now 2 years. In the Simon Simplex collection, primarily work between our group, that of Matt State, and that of Mike Wigler, we managed to sequence 10,000 exomes. I know there was a parallel effort at the ASC here with Joe Buxbaum. In our study, we characterized all 2,517 families, and we validated 2,162 de novo mutations. So by this time, after complete sequencing of 2,500 families, we had 26 genes with a recurrent likely gene disruptive event, 0 in siblings, and 70 that had 1 LGD and an additional de novo missense, as well as 83 genes where there were 2 or more de novo missense. So these are obviously outstanding candidates for being autism, significant autism risk factor genes. The other thing which I like to make a point of is when we did this analysis, we were all running kind of older versions of GATK, and I had a student basically spend three months recalling the data. So taking the data at that time, which for us we have to stretch a little bit, and we have to go and work on the cloud to do some of this. I gave him a limited budget, $32,000. He'll always remind me it was $32,000, not 1 penny more. And his job was to recall single nucleotide variants and copy number variants in the exome data, do it within 2 months, and keep under budget. And he managed. Nick Crum was his name. He managed to get it done with $2,000 left to spare. And essentially from that analysis, he picked up another roughly 1,000 validated de novo variants that we had missed on the initial call sets that had been generated. And that was just by running freebase on top of GATK. So that wasn't bad, because from that came 21 new recurrently hit genes based on a kind of reassessment of the data. So this is a probably important message for a lot of students and postdocs. It's good to reanalyze. It might seem incredibly boring, but in fact, there's power in that. You can actually get much more out of your data. So at the end of this, we had about 200 candidate pathogenic, putative pathogenic genes to go forward on. In this study, we had ability to go back and now look at the different types of mutation types. Now we had so many disruptive events. We could look at different types of nonsense or likely gene disruptive, including nonsense and splice sites. And this is just to show you the rate per child. So this is taken from the ISFOP paper. You can see there's a clear 2-to-1 consistent pattern that we see in terms of de novo mutations, the rate of de novo mutations that kill genes in cases versus controls. This, to me, was particularly interesting. You can see that there's no difference in terms of the synonymous rate of mutations. But if you look at the missense, you can see there's a subtle and significant effect. You see a slight excess of missense, about 0.01 in terms of significance. So we estimate from this, based on the rate of truncating mutations in probands versus siblings and that differential over the total rate, that about 42% of the likely gene disruptive mutation events that we see are risk factors, if they're de novo, for autism. Importantly, we went back, and we spent a lot of time doing this, we went back to address the question, is it possible that a de novo likely gene disruptive event was occurring with the other allele being disruptive, and we just hadn't seen it because it wasn't de novo, so in other words, a recessive model for the disease, and we found no occurrences of compound heterozygotes. So this goes back to that heterozygous model of disease. A lot of us had been thinking, and there were reasons to think about it from certain Mendelian families, that would be an autosomal recessive model. And the data suggests that it's essentially not. It's an autosomal dominant model. Taking out one copy of these genes is enough to provide you significant impact and increase in risk for autism. So that's our estimate for de novo likely gene disruptive events. And this is our estimate rate of missense in probands, as I mentioned, is slightly higher versus the unaffected sibling. You can do the same calculation and you can estimate that about 12% of de novo missense have something to do with autism risk. So that means the haystack is bigger, the needles are smaller. That means 90% of the time you see a de novo missense in an autism family, it has nothing to do with the disease. But 10% of the time it does. And these are obviously, in fact, when you do the math and you ask the question, how much of each of these contributing to the total number of patients in terms of risk factors, this is about 9%, this is about 11% of the patients. So combined, it's about 20 to 21% of the risk is in de novo, but more than half of it is coming from the missense, even though we have less power to actually prove the pathogenicity of any given gene.
Do you ever look at whether the genes are expressed in brain or not as
Yeah, we do, and others have done this too. A lot of the genes, but not all of them, show pretty strong expression patterns in the brain, particularly early in development. We're talking early, 7 to 12 weeks of development. There's a particular spike on subset of genes that are there. And then there's a whole set of genes that are actually expressed postnatally. At least they reach the highest levels postnatally. I'll show you some of those. All right, so this is just the combined from the Simon Simplex. These are the list of all the top genes. So CHD8 is a gene we identified in 2012, and I now had 7 patients from it. Nothing seen in terms of missense at that time, and nothing seen in the unaffected SIBs. And this is the top ranking genes. And this is for missense. The greatest number that we saw was for SCN2A in terms of de novo missense, and then it continues to go down. But you can still see, even though we have strong statistics here already, by the time we get to 3, for sure, that this is now going to be a risk factor, most of the genes that we're seeing are seen once or they're seen twice. And you can't be absolutely sure that you've identified a risk factor. All that you have is these aggregate statistics. So this is the fundamental problem with an individually rare, collectively common disease, is you need to screen not hundreds, not thousands, but you need to screen tens of thousands of patients. Because what a family with autism wants to know is when a child is seen with a mutation in, let's say, ANK2 or INC or 6B, is that the reason my child has the disease? That's fundamentally where we need to be in the field if we're going to provide useful diagnostic information coming back to the families and ultimately therapies going forward. So this was the problem. The disease, based on our estimates, was 500 to 1,000 genes. In every family, there was a different gene that was contributing to the disease, and the chances that you saw the same gene be hit twice was, in fact, very rare. So you could be a clinician seeing patients for 30 years and may have never seen the same genetic cause of autism more than once or twice. in your years of practice. So we need two things. We need to be able to gather not thousands, but tens of thousands of families for essentially doing this type of analysis to actually prove the pathogenicity of individual genes. And the second thing we need a technology that can do it cheaper than exome or genomes. So while exomes and genomes are now cheap, or were cheap even back then, exomes at that time cost us, I think, $1,500, and now genomes are at that level. If you have to do tens of thousands, even Jim Simons doesn't have that kind of money, or at least he doesn't have that kind of money for us, right, to do that type of sequencing. So we need to have a technology that can zoom in to the specific genes of interest and in fact, find the mutations. So I had the good fortune of being right next door, actually one floor away, from Jay Shanduri, who had just come to University of Washington from George Church's lab. And he had been working on this technology when he left George's lab, which is called molecular inversion probes. For those of you who are not familiar with it, it is a wonderful technology. It's essentially glorified oligonucleotides with complementary arms, very akin to padlock probes, targeted to your genes of interest. So this could be your favorite autism or your 50 autism genes that you want to do. You design thousands, typically on the order of 3,000 to 4,000 of these inversion probe assays. And the target distance here is only about 120 base pairs. These are highly multiplexable. And what you can do is, if you have high-quality DNA from patients, you can make 96, 192 microarray dishes, put your gamish of oligonucleotides or MIPS into these assays, throw in a DNA polymerase, ligase, add a barcode, pull samples, and sequence. And in work that was done part by Jay and in part in collaboration with the postdoc that we had, Brian O'Rourke, in our lab, could show that you could routinely sequence 192 samples for 50 to 60 genes with high specificity, high sensitivity, capture information on 92% of the exons, and you could do it cheap. And by cheap, I mean we could sequence 50 genes in one sample for the reagent costs and amortized machine of about $12.50. So that's exactly what we needed. Not hundreds of dollars or thousands, we're talking $12.50 to now resequence tens of thousands. The second thing we needed tens of thousands of patients. And so the way we did this is we set up a network. I spent way too much time between 2014 and 2012 in airplanes flying to these different places to meet with investigators to build a cohort, which we've called the Autism Intellectual Disability Network, or ASID. We actually went back to 10 of the 13 centers that had contributed samples as a part of the Simon Simplex collection. This required actually 13 IRBs for each center and an Uber IRB to actually break the code. But we were able to do that. And as you might imagine, most families wanted to be recontacted. Because this idea that families don't want to be recontacted is false, at least in terms of cases with autism. They want to know what we're finding. And the important part of all these families and these collections is these were all consented not only for doing genetic testing to discover the cause of autism or intellectual disability in these families, but they were also such that if we found something, we can go back to the family and we could explore the phenotype working together with the family. And that was the really important piece to what I'm about to show you, was the recontactability of these families once we discovered potentially pathogenic events. Because in our mind, and we knew this from CNVs, if we can go back to individuals conditioned on the same genetic type of mutation event, and if that mutation is truly contributing to the disease phenotype, we would expect there to be more similarities in the context of that family as opposed to looking at generic autism more broadly defined. At least that was the test. So this, for those of you who are interested, these are all the colleagues that have participated in this. Unfortunately, we don't have 18,000 at any given moment, so some sample collections are very limited. So we've now, I think the number is about 15,000 patients have been targeted resequenced for a full set of about now over close to 250 genes that were essentially discovered from the exome sequencing projects. So this is a little bit out of date. So this is data from 155 genes unpublished, looking at 92% of the exons, 8,000 of these, I said 155 genes in 8,000 patients. When we did this analysis, we now had 65 genes with now 4 or more likely gene disruptive mutations and 0 non-affecteds. These are about as slam dunk as you can get in terms of actually genes related to autism. So we're very confident about these genes. And then we have other sets of genes, 92 of these, where we have an excess of severe nonsense mutations as well as likely gene disruptive events. What I'm showing you in this fairly busy table is all the data that you see in the second column is the data that we discovered from the new sequencing of patients. And the numbers that you see here is basically a database. It's actually up to database 9 in our group, which includes Diosiphoff and the DeRuby's paper from the ASC in terms of the number of de novo mutations that were actually identified in that database. So combined, the total is the number of patients that we now have with a specific mutation or a set of mutations in a specific gene. So disruptive events. So in many cases, we're now double digits, or we're getting close to double digits, which now allows us to say, let's bring the families back in, because we now have confidence. And the clinicians that we're working with, when we show them the data and we say we got 8 likely gene disruptive mutations in gene X, and we got nothing in controls, they say, good enough for us, let's bring them in, let's study. So we make through most of the IRB hurdles by really doing this kind of statistically based analysis first before going forward. So what are we finding? So this is what we call our genotype first based approach, because we're actually discovering the families based and grouping them based on the same genetic lesion in those families. So this was our first and probably one of our most significant hits, CHD8. It's a chromatin helicase DNA binding protein, 8. So there's a family of 9 of these. And these are some of the families that we brought into Seattle, and there's actually a family here also from the Netherlands that was brought in, the patients. And the first families that were brought in, we actually saw striking similarities. They were unrelated families, de novo mutations in all cases. We saw, for example, that a diagnosis of autism was made in about 90% of the cases. Only half of them would have had intellectual disabilities. This is not just an intellectual disability gene. This is an autism gene. 80% had macrocephaly. So you can see this, kind of these large foreheads. These are just based on measurements of head circumference at various stages. Interestingly, 73% of these kids had gastrointestinal dysfunction, and it was particularly severe. So you often hear of GI disturbances with kids with autism, but in these cases, these kids would go for like 3 weeks with diarrhea, followed by 3 weeks of 4 weeks of constipation, and over again until about the age of 10. And then the symptoms, either by some type of remediation or maybe because they grew out of it, quote unquote, the symptoms would more or less disappear in these families. So it's a very specific feature. And this was significant when we looked at the entire SSC. And the other was sleep dysfunction. The families came in often and would tell us that their kids would often go sometimes 3 days without sleeping 1 wink. And I remember one mother telling us that she said she could not stay up with her son anymore, so she ended up locking the door at night, kind of locking it from the outside, because she was afraid he might wander away, because she always wanted to be awake when he was awake, otherwise some bad things would happen. And to me, this was, even though this hasn't reached significance yet, we think when we get to the numbers, the larger numbers, will actually get significance. So sleep dysfunction, GI disturbance, macrocephaly, and you can see from these statistical models, looking at just even a simple Fisher's case control, we have significance. And if we look at a locus-specific significance for de novo mutations, pretty much every mutation, almost every mutation that we type is de novo, even that we're going into these families and we're discovering them not based on de novo, just based on the class of mutation. So we had a chance to model this. There was no mouse model that we could at least access. So we did work in part with a postdoc in my group as well as Nico Katsanis. We knocked out this gene in fish, the zebrafish. We first started with morpholinos, and then reviewers made us do it with CRISPR, because that was now the metric for essentially this type of work. And in our analysis, we saw a consistent increase in distance between these eyes. This was essentially kind of the equivalent of head circumference. In fish, you measure the distance from these eye cusps. And we then had to subsequently go back and show by marker analysis that there was, in fact, an increase in the number of cells of specific types, particularly cortical neurons. So we had a significant, for several markers, there was an excess of cortical neurons that were in the fish, which presumably is analogous to the macrocephaly you're seeing in the patients. The other thing which is pretty interesting, we also looked at the GI disturbance in the fish. And what I'm showing you here is staining for an enteric neuron, so an antibody that stains the enteric neurons in the fish, showing you the control and the morpholinos, and we saw this also with the CRISPR, is that they had a significant reduction in the number of enteric neurons, either representing a migration or a proliferation defect, but opposite of what we were seeing in the developing brain. So in the brain, we have an excess in cortical neurons, and in the gut, we actually have a deficiency in a number, or a depletion in the number of enteric neurons. And you can look at the paper. This has been published, but we've also showed that if you do a microgravage assay and you stuff these fish with kind of fluorescent beads and you monitor the progression of these beads through the gut, the beads take longer to get through the gut, suggesting that these fish are having a problem because of probably this enteric neuron deficit of getting essentially simple digestion, in this case of using this microgravage assay. So all told, we feel pretty cool about this one. It's one that we found the first 2 recurrent hits in 2012. We now have close to 30 families. We have a phenotype. We have a model. There are mouse models being built, and it seems to be a high-impact gene that's almost as necessary and sufficient, at least that we can see, for developing the disease. So the second one that I'll show you, so this is a dual tyrosine phosphorylation regulated kinase, 1A. We found hits in it. It's an interesting gene. It's been studied actually for many years because it's one of the 2 genes in Down syndrome patients that people believe to be responsible for the neurocognitive deficits associated with trisomy 21. So when we started getting de novo hits, like the gene disruptive events, so these are now taking out 1 copy, so the opposite of Downs, we said, well, we got to focus on this one and see what we can learn from the families. I'm showing you 3 examples of children with the disease. So once again, this is highly significant, both by the de novo and the Fishers. These kids are, and as opposed to macrocephalic, are microcephalic, so 100% are microcephalic clinically. 90% of them have late onset epilepsy, so we think this is going with the disease. In this case, remember, we're ascertaining patients based on developmental delay half the time versus autism. 83% have a diagnosis of autism. And what clinicians are convinced of, and I've seen now close to about 20 of these photographs, the kids have a very specific set of features. So they have this microcephaly, but they also have this kind of very specifically pointed chin, long nasal bridge. Some of the effects are pretty pronounced in some of the more severe kids when you see it. So there's clearly a dysmorphology going with this one. I can tell you that many of the children that in fact are adults that look normal, they've had to have reconstructive surgery, in particular removing teeth during development because there's not enough space in the developing jaw for all the teeth to actually erupt. And so it's common in these families to actually have teeth extracted as they're developing. So in this case, a mouse model already existed. It was work that was done previously because of the association with Down syndrome. And people had shown that even heterozygous mice for the knockout, in fact, had reduction in the brain, particularly the mesencephalic tectum and medulla oblongata. And there were behavioral deficits, including delays in pre-weaning, delays in reflexes, and increased anxiety, which appear to mimic at some level what we're seeing, at least in the patients. Third example is work that we did with colleagues. It's a gene that we and others I don't think had implicated in autism prior to the exome sequencing in any stretch of the imagination. It's an activity-dependent neuroprotective peptide, and it turns out this is a gene that in fact is thought to protect the brain from certain neurotoxins. And it's been studied by people working in the field of Alzheimer's, particularly the derived peptide, as a potential therapy for Alzheimer's. So we found this gene in patients with autism. Everything that we've typed was de novo and highly significant. These kids have ID as well as ASD. They have very severe loss of expressive language, which was identified by most of the clinicians around the world early in their clinical reports. And they don't have either macro or microcephaly, but you can see from these pictures, they have this kind of unusual frontal bossing of the forehead, as if the frontal cortex is expanded in these particular children. And in fact, there is data, it's still fairly minimal, but there's data being collected that these kids have a significant MRI abnormality associated. So not micro or macrocephalic, but a very specific effect that's being seen in the prefrontal cortex. This mouse, when essentially mutated, it's essentially lethal in the homozygous state. It results in essentially failure of the neural tube to close and early death. And heterozygotes mice have a deficiency in glial pathology as well as reduced cognitive function. The last one I'll show we just published a month ago. This is a gene that is loosely described as a transcription factor. It's called POGZ, which I have to say is a better name than Pogo transposable element-derived protein with zinc finger. It's a gene that, again, we would never thought to have gone after. Working with Tjitske Kleefstra in Netherlands, we now have identified 24 new likely gene disruptive events. We found a total of 25 patients thus far that have essentially likely gene disruptive events, and essentially 0, or maybe take that back, there's like 2 in the exact database that we found. These kids have mild intellectual disability, but they have a phenotype that some of the clinicians said this is almost the anti-autism phenotype, because they were often happy and friendly and extremely compliant and want to interact with people. These kids are often still very shy and are well organized, with a tendency, as some clinicians have written, to be obsessive compulsive in their kind of responses and interactions. So a very interesting behavioral phenotype that I think is going to be worth following up more. Compared to the SSC, they are both microcephalic, hyperactive, and they have what is described as aspecific vision problems. So that these kids have a real problem, in fact, in terms of seeing depth and seeing, but it's not the same type of defect in all the kids, but there's an effect that's going on with the eye. And you can kind of see from these pictures, there's a tendency toward obesity, and there's an issue of kind of hyperphagia, so a feeding problem. So we now have a total of 25 LGD mutations compared to 2 in EXACT, which is the control database. And from that, we can use it to calculate an odds ratio. And this is what I love for all my stats friend. The odds ratios here are 35.8, with a range of 8.9 to 311 in terms of the confidence interval, and a penetrance of 65.9%. This is where we need to be for the disease, because we can tell patients now, families, when a POGZ de novo mutation is occurring, we know we have power to say that this is likely the cause or will be the cause of disease. And I know that's a loaded, cause is always a loaded thing in genetics, but these are the types of numbers that we're going to need going forward. So what do these genes all have in common? And many of us in this room have worked on this problem. These genes are not mutations randomly distributed in genes all over our genome. They are genes that have related functions. What I'm showing you here is a PPI, or a kind of a, we consider our core protein-protein interaction network, where the size of the circle represents the relative number of likely gene disruptive events that we've seen in autism patients. Red is likely gene disruptive, and blue is actually de novo missense mutations. And anywhere you see a joining line, that means there's good evidence in the database that these proteins actually directly interact with one another. So in our mind, there are 3 pathways: synapse function, which has been implicated many times in many studies from the CNV work from Steve Shear and others early on. Wnt signaling, which we see what we think is convincing evidence that this is involved, and also see chromatin remodeling. A lot of genes involved in modification of histones and remodeling of chromatin more generally. And in fact, if we took at this time, and this is 2014, if we took all the likely gene disrupted de novo mutations and we threw them onto this map and said, let's look within 1 to 2 steps of these kind of core genes, this is something like 65 or 70% of the de novo mutations that we're finding kind of can be transposed on there. And then you'll see certain things like things that are not necessarily Wnt signaling, but this is the E3 ubiquitin ligase network over here that's starting to pop up. So there's tremendous power in the network-based approach. I was not a believer in 2012. Brian O'Rourke, among others, have convinced me that this in fact does work. We can have predictive power at the network level to identify even new genes with a higher prior if we've only seen 1 severe de novo missense or de novo likely gene disruptive event. And why I think this is so important, and the data are still early, but we know that genes that are operating within the same network or the same pathway, those kids have similar or opposing phenotypes. So for example, the micro-macrocephaly phenotype, we're seeing predominantly over here in this part of the network. And when we go to these path networks, we do not see, when we look at these patients, we don't see this macro-microcephaly kind of distinction going on. For those of you who are interested, the synaptic plasticity, this is the gene family that we were talking about, a set of genes, I should say, that is associated with postnatal. So these genes reach their highest peak in the first year postnatally, while most of these genes on this side of the fence, including over here and over here, these are ones that are expressed particularly early in development. So 7-10 weeks post-conception. All right, so that gets us to exomes and the power of exomes. But obviously, if you combine the CNV counts in terms of risk, and you add the de novo missense and a de novo like gene disruptive, where's the rest of the mutations? We want to get to essentially 100% of the genetic risk. That's what geneticists' job would be in the case of autism. And where to look is obvious. We want to look for inherited mutation, and we want to look at non-coding regulatory mutation. In other words, look at inheritance of mutation events as well as areas that are likely going to have an effect in terms of regulating the expression of genes. So when I made that student for $30,000 go and recall all the 10,000 exomes, it wasn't just to discover additional de novos. It was specifically for this question, because what we wanted to be able to do is go back and have no under-ascertainment in the parents. We wanted to be able to look at transmission of every particular variant that we discovered. And because we had 10,000 individuals, 2,500 families, we could say something about the minor allele frequency of the types of mutations that we would see transmitted. So what I'm going to show you here are 4 different categories of very low minor allele frequencies from 0.2% to private. So private means that we've never seen this mutation in another family or in the database. So this is something that we think is private, even though it's being transmitted in the family. And the rest is just based on the allele frequencies that we see within the exomes. And I'll break out missense in gray and likely gene disruptive in red. I think this is really cool. So when we did this analysis, the only place we saw significance is in transmitted variants, which were private. And we only saw it for likely gene disruptive. In other words, it doesn't matter if we use a minor allele frequency of 1 or 0.1%, we don't see a significant odds ratio comparing the affected versus the unaffected until we get to private mutations. In other words, affected individuals within a family are more likely to inherit likely gene disruptive variants from a parent than the unaffected in the context of that family. And if we take David Goldstein's RVIS score, which gives you a sense of how tolerant a gene is to mutation. So a RVIS of 1% means a highly conserved gene that doesn't get mutated in the human population. 100% means that you see lots of mutations in this gene all the time, suggesting that this would be under stronger selection. You can see that the odds ratio, really when you get to an RVIS score of about 20, begins to peak. You see this kind of rise in this. So this actually tells us something pretty fundamental. Private mutations are transmitted. In aggregate, they're being more likely transmitted to children with autism and in genes that are intolerant to mutation. So we can now put metrics on this based on this kind of aggregate analysis. In terms of the phenotype, this I think is also particularly interesting. So a lot of people have criticized our initial studies, both from the ASC and SSC. A lot of my colleagues in Europe in particular says, all you're discovering is intellectual disability genes. That's what they've told us. Even though they were diagnosed kids with autism, a lot of those kids that were, it's true, that have the de novo likely gene disruptive events have also intellectual disability. Not that that's a bad thing to be discovering those genes, but they say you're not finding autism in individuals without intellectual disability. When we look at the private LGDs and we break them out based on IQ, what you can see is that we see significance for less than 70 in terms of IQ, but we see the strongest signal coming from these kids between 70 to 100. So it doesn't mean there's not a cognitive depression, but what it's telling us is that in terms of these inherited mutations, these may be more likely relevant to essentially the families in which you have autism without intellectual impairment. And then the last thing, which comes back to the whole gender issue, we asked the question, did it matter if you were a boy and a girl in these families? So this is something like PAR, it's called attributable fraction. So it's trying to estimate the number of patients that you can explain for by this type of mutation. So we're looking at inherited, likely gene disruptive events. Here's the aggregate number, whether you're male or female. Here's if you're a boy, and this is if you're a girl. And you can see the only significance that we see is if you're a boy. So 5.15% of boys that we see in our data, we estimate in terms of its contribution to that fraction of the population that we're looking at, and essentially nothing for the girls. Got to keep in mind that these are pretty wide intervals yet. That's because the sample sizes are still relatively small. Just to give you a point of reference, for de novo mutations, although Mike Wigler would say different, we don't see a big difference in terms of the attributable fraction. De novo mutations are affecting boys and girls in terms of autism, at least based on the samples that we have. So then the question is, does it matter if you inherited this event from your mom or your dad? Right? That would be the next question. So this is inheritance coming from mother. And you can see the attributable fraction of autism actually spikes and increases even more, 8.32. So 8% of autism, this may be a relevant risk factor when transmitted from mothers to their sons. If you look at essentially transmission from the father, you see no effect. So remember that thing I was telling you in the beginning about CNVs? And 80% of these things, or 60% of these things coming from moms to their sons. We now see the same effect in terms of likely gene-disruptive private events being transmitted preferentially from mother to their sons. Consistent with the idea that mothers are more likely to be carriers, and we know that fathers are more likely to be contributing through their germline the de novo events to the offspring. So we have this kind of, I think now beginning to form a genetic architecture model for the classes of variants or mutations. We know that large CNVs are important, and when transmitted, they come preferentially from mothers. And I think in the newest numbers that we calculated in terms of large CNVs, it's like 70% or 74% of them are coming from mothers. We have excess de novo gene disruptive events. They originate preferentially in the paternal germline. I didn't show you that data, but they're obviously contributing a significant risk. Then we have excess private likely gene disruptive events. When transmitted, come preferentially from mothers to their sons. The last part is just essentially full genome sequencing. So now that we're kind of exhausting what we can get out of the exomes, we and others have turned to full genome sequencing. We published a pilot study on this beginning of this year, just showing you the first 208 genomes from families from the Simon Simplex collection. So there's a total of 53 families, most of them quads. And this is particularly important because when we got these whole genome sequences, we had oodles and oodles of variants. And the question is, how do you begin to look for any signal in all of this variation that you see? You can sort by frequency, you can sort by all types by size. These are all important considerations, especially for CNVs. So what Tashelle Turner did, and I think makes total sense on this, so she said, well, we know that dosage sensitivity is important for autism. That's a given. Let's look at regions of the genome that have DNase hypersensitivity sites, specifically from the CNS, from the fetal CNS. So these are genes that would be, or regulatory elements that are potentially important early in development. And let's look only at genes which we have a prior for autism risk. Okay, so those are the conditions. And when she did this, she saw, and this is a very modest p-value at this point, but remember it's only 53 families. These were families that were negative by exome, negative by CNVs. She saw a transmission disequilibrium coming, once again, to the affected individuals from mutations, particularly CNVs and de novo events that were actually occurring in DNase hypersensitivity sites in patients with autism compared to the unaffected CIVs. So I'm excited by this because I think, obviously, this is the first thing we want to replicate as we start to sequence, and we are sequencing and analyzing additional genomes. Because if this is true, we've identified a new class of risk. These are now things that are not necessarily in the protein coding space, but they're actually in the regulatory space. And they're actually disrupting, specifically in genes that we already know are implicated in terms of autism. And so here's some examples. DSCAM is a gene that has reached significance. Here's an example of a deletion that we identified, 14 kilobases transmitted from the mother to her son. Here's a triplication of a gene. A paper just came out. We know this is a gene implicated in autism. We have a duplication that involves DNase hypersensitivity as well as H3K27 acetylation signatures that is transmitted from the mother to the son. This is what all the families that we think we've identified potentially pathogenic events in. So these are all negative by CNV, negative by exome. We think we have close to about 8 families where we in fact have these types of mutational events that are segregating. So this is this DSCAM. Here's another example. You can see that you have a lot of boys once again, but you can see that when you look at the girls often have multiple events that are coming into play. So for example, this girl here has a deletion that takes out part of just adjacent to the gene that's known to be in autism, SCN2A. She inherited this from her mother. She has a 12 kilobase deletion from ARID1B, which is a high impact neurodevelopmental gene. She inherited that from her mother, and she got a calcium channel mutation coming in from her father. So what do you do with this stuff, right? You get lots of these kind of potential regulatory elements, and then you get a reviewer and an editor that says, well, why don't you just validate them? And this is where it gets tough, because we're not talking about just looking for a transcript or the absence of a transcript. You're now talking about proving that these are regulatory elements. We need methods that are going to be more high throughput to do this in a systematic way. We teamed up, for example, to show the reviewer and the editor, for example, that this works. So teamed up with Andy McCallion, we took this 14 kilobase element, we looked at all the DNase hypersensitivity sites, and he took essentially 9 of these and he made transgenic constructs or tested the constructs specifically in zebrafish. These are transient, not transgenic. And he could show that essentially 9 out of 9 of these was actually driving expression, specifically in the CNS. So he was actually super excited because he said the chances that I would draw, he said 9 DNase hypersensitivity sites from one region and have them all drive individually expression in the CNS, he says makes this a super regulatory region, right, for thinking about regulation of this gene. I'm not sure if that's true or not, but that's based on his empirical data. But I think it's kind of cool for those of you who are interested in regulatory sequence as opposed to essentially coding sequence, because it may be that autism is the perfect system to actually detect such events, because in order to have a significant effect to lead to autism, we may be picking up these regions that are really jam-packed with regulatory sequences to bring the dosage of a gene down by 50%. All right, so in summary, I've talked about really 3 classes of mutation. I've talked about this de novo, as close as we get to necessary and sufficient type of mutation. These tend to have a higher impact on IQ, and these are the large CNVs. The de novo, likely gene disruptive events, which we think account for about 30% of the risk, of the genetic risk of autism in families. We have the class 2, which I talked about from the 2015 paper in Nature Genetics. These are the ones that are being transmitted. And when they're being transmitted, they're likely gene disruptive. They're private, so they're exceedingly rare. They're as rare as you can get other than de novo. And they're being transmitted preferentially from mothers to their sons. These tend to have, at least from what we've seen, tend to have milder impact in terms of IQ. And the last, which one has to say be very careful on this yet, because the numbers aren't big enough, is really the small CNVs, particularly de novo mutations as well as inherited mutations that disrupt regulatory elements outside of coding regions of genes. And these ones, we think it's going to be a tough model, because if these are supposed to be primarily monogenic, this type of event is going to be more or less oligogenic. We think you're going to have effects from 2 or 3 genes to perhaps get a strong effect with respect to disease. And hopefully I've convinced you that this genotype first-based approach for breaking down the subtypes of autism is actually working, but we need more samples, more patients, more involvement of families to make this work. So we've come a long ways. I'm definitely a glass half full kind of guy, not empty. I think we've made, and as a community, we've actually kind of moved the dial in terms of thinking about the genetic basis of autism, with a lot more genes and a lot more types of mutations that now can be implicated. I think we have a lot of hope, and that's probably where we are. Obviously, there's immediate benefit for the field in terms of diagnostics, being able to tell a family this is why your child has autism. One thing that I've seen from these family interactions, and one thing that most researchers don't appreciate, I think, is that once you've identified the 10th and 11th family with a CHD8 mutation or an ADNP mutation, they connect with one another. And the type of real practical support that these people are giving each other actually beats all the therapies that we'll develop over the next 10 years. Because when I've heard many of these stories where 45-year-old mothers are now talking to 25-year-old mothers with a kid with CHD8 about the things that worked and didn't work with that specific type of mutation. And the parents are incredibly savvy. And I think that's something that the clinicians need to heed, is that they become better experts than the clinicians and the researchers, because that's all they think about is that type of disease. So while there may be hundreds of different genetic subtypes, I think ultimately they're going to come down to the proteins and then the networks, and the therapies will come and be testing against these networks. So I'll just leave you with 2 things, and I don't want you to get anything from this, but just to say where my mind is right now. We've gone through a transition in our lab of doing everything in-house to now actually thinking about ways to analyze massive amounts of data. And so everything we used to do, we were pretty proud of the fact that we built our own compute farm, 2,600 CPU cores. We had 3 petabytes. We could easily analyze genomes worth of data as well as exomes. That's fine. But now we are moving forward to actually analyzing hundreds and thousands of genomes. And that is not fine anymore. At least in our lab, there's not enough compute space, physical space, enough cooling, nothing that we can actually do to actually build essentially and analyze these genomes in real time. So what we've done is we've started to create a hybrid model with everything indicated in green are the things that we are doing with respect to the new thousands of genomes that are being sequenced from part of autism as part of a collaboration with NYGC, what we're doing on the cloud. Everything that you see in blue and in some level here, the beige, are things that we're taking off the cloud and doing locally. And this is kind of the complexity. So for CNV analysis, you can't just run one CNV color. All of you should know that by now. You have to run at least 3 or 4, and even then your sensitivity is in the toilet. But you've got to run more than one. You have to have a read depth color, and it's more computationally expensive than some of the other methods. So what I told my postdoc the other day is, if you would have told me 5 years ago that I would have to be monitoring this level of kind of management of data analysis, I would have said no. And the part that actually freaks me out the most is I monitor also exactly how much each sample costs. So I know, for example, that the QC metrics on the cloud are costing me $1.32 per sample, and that this analysis of rerunning Freebase in addition to GATK is going to cost me $7 per sample. And if I really want to run my software that we've developed in terms of multi-read mapping and actually call copy number variation with the best sensitivity, I have to invest $23.48 per sample. And a lot of you are saying, well, that sounds cheap. Yeah, it sounds cheap until you start talking about tens of thousands of genomes, right? And each of these has time associated with it. And so this is something we got to get much better at. We're just starting to suffer this right now. It took us 3 months of racking our brain and realizing that the cloud people, whoever they may be, these people that you're assigned to work with when you start working with Google or Amazon, are about as clueless as we are. And even their software doesn't actually work the way they think it does. And so we've actually learned more on our own. They've asked us to rewrite some of their fundamental scripts, which I just think is absolutely ridiculous. But data management and the price structures are something that is a challenge we've got to face. Another challenge, and most of you people probably don't appreciate this because you think that when you sequence a genome at 30x sequence coverage from Illumina, whatever the platform, Illumina technology that you're doing, iSeq or X10, that you think that's good enough. I hate to tell you this, but we've now sequenced genomes with alternate technologies like PacBio, and I can tell you that we're missing 80% of the structural variants that are in a given genome. 80%. And the reason for that is that insertions and deletions, especially when they get a little bit bigger than 50 base pairs in size, are non-randomly distributed even in local space. So this is just to show you, everything in dark black and dark red was from a paper that we recently published. It's just to show you that, in fact, most of the genetic variation, particularly when you get below essentially 1 kilobase in size, is new in a genome when we sequence with PacBio. Now, obviously, PacBio can't be used for autism sequencing or clinical grade sequencing, because this genome cost us $80,000 to generate. So just try to factor that into your calculation of price, right? But this is where we need to move as a field. We need to be moving to a place where we can comprehensively understand all forms of genetic variation, because I will put money, and I will bet anyone, that we're going to find disease-causing variation, a lot of it, specifically in this space of below a kilobase. Because think about it, delete or insert right over a regulatory element, 100 base pairs, yeah, that's good. You will have an effect. But if you can't detect it, you're in trouble. And then the last thing, I thought I had one more on this. I think the last thing I was going to say is, as a community, I think the other challenge is coordinating the efforts. So the fact that there's an ASC and an ASID and that someone is doing something in Europe, we now know, and we've known this now for a couple of years, that we need to connect among all of us. We need to be able to coordinate these activities at a higher level, and we need to be able to recontact the families. And I think that's a part where the different countries have different rules, and we're struggling with this. But when we've worked with the families, this has been the part that they want. They want the information back to them. And you say, well, why? Why would it be important for them? Well, if you think about Rett syndrome, you think about Fragile X, the families, once they connect with one another, they will start making their own decisions whether they participate in a particular therapeutic or a phase 1, phase 2 trial or not. Those are to be the first people for any of these therapies that begin to be developed, especially when they're conditioned on the genetic lesion, that will line up for the therapies. So even though I still think light years away from really practical therapies that will make a huge difference, there are things that will actually provide, probably efficacious results and directions for future research. And I think that's why it's so important as a community that we need to network at a different level than what we've already been doing in terms of the consortia. All right, so I've gone over, but I want to acknowledge particularly to Shel Turner, who allowed me to show you some of the unpublished data that she had. A lot of collaborators around the world, in particular some of the groups, particularly in Europe, we've worked closely with. The funding agencies, and most importantly, the patients and their families, without whose contributions we can't do this type of work. So we are eternally grateful for their involvement in the research studies. All right, thank you.
Well, thank you for a fabulous talk. From my perspective, well worth staying. So are there questions? I'm sure there are. While people are leaving, maybe I'll start with one of the 12 that I thought of while you spoke. So people inherit, they're common, they're rare, they inherit lots of things from their parents. So all of the variants that you've talked about, and I understand exactly why you do this, you want to find the strongest of each class. So you're looking at those alone. But your pie at the end still has 50%. So what about when you leverage that back and look at combinations? Does that help you make more sense of the missense, for example, the de novo missense is the other, that sort of integrating for an entire person. And I'm not going to ask this question now, but the reason I partly say that is I obviously know that many of these cause schizophrenia, are associated with adult psychiatric syndromes also. So what are you telling patients, and does that help you know something about the phenotypic spectrum in some of them now that you have 30 cases, for example?
Yeah, it's a great question. It's all of this is kind of triage at this point. So we're saying we want the most severe mutations, we want the rarest of mutations, right? And we know that only gets us so far. I've seen some early data. So from other studies, we know that there is a polygenic component. So it matters upon which genetic background these mutations exist. So in other words, it's another way of saying that common variation is going to be relevant, and it probably is relevant, I think, more in terms of the expressivity. So whether a child will manifest as an epileptic or a severe intellectually disabled kid or as maybe even a normal individual, right, without any type of obvious clinical. The question regarding, full integration, the way we're doing it is, like you said, we're starting with kind of the most penetrant and working backwards. So one of the things that we're starting to do is we're starting to look at missense mutations now within families that are below essentially our thresholds initially. So we had very severe missense is how we categorized beginning. So we had a CAD score cut off at 30. And I can tell you that when we actually take that filter off and we go to genes with a prior now, we're starting to see a significant enrichment of de novo missense mutations that are actually at a lower CAD score. So one that all the computational people would say, that difference wouldn't have had an effect. And so I think what that's telling us is much what you're getting at, is as we go to these less penetrant mutations and less penetrant effects, we're going to get genes that are contributing, but it'll be a gene here, a gene over there, a gene over there. And that's where we have to kind of, I think, integrate the oligogenics first. So I talk to Mark Daly about this all the time, and Mark says, well, you got to do the polygenic stuff. He said, you're spending too much time in the monogenic side. And I said, yeah, but I said, polygenic is not going to get me to the genes. I said, oligogenic, I have a hope. And I believe there's a big chunk of space in oligogenic, and oligogenic here would define as 2 or 3 genes in a given patient as opposed to, let's say, 20 or 30 genes. And so I think that's where we want to do this type of analysis. And I can't overestimate the value of having genetically and statistically robust footholds in this field. And the fact that we go on and we calculate these aggregate statistics in the beginning was interesting, but not really the foothold that we needed. We needed to be able to drill down to a specific gene and say yay or nay. And in those lists of genes, that some with even 2 de novo mutation events, when we've screened 18,000 patients, we've rejected some of those events. And even when people said there would be a 95% chance that that was actually going to be a gene, but when you base it on small numbers, it can go any way. So I think the first part is really kind of getting the family numbers and patient sample numbers up, which is something that pretty much every group, including the schizophrenia groups, have shown, right, over the years.
Zaina?
Hi, I have a question regarding the recurrent mutations that you found in specific genes. How much did they overlap with the CNVs that you found? I mean, are they completely different regions, or are there some overlap? And if there's overlap, is it the same? Well, I assume it's different people, right?
Yeah. So there's some of the genes that totally are embedded within the actual CNV. So we know we built a morbidity map across the human genome based on CNVs in kids with developmental disabilities. So we have for every base, we have a kind of a prior expectation of dosage imbalance and its relevance. It doesn't necessarily mean it's significant. We just have this, we know, and there's some that are significant. I would say in about 30% of the CNVs, exome sequencing has been helped us to actually refine the most likely gene. But for a lot of other ones, there is no single gene underneath it. And it may be for some of these CNVs, it's not the fact that you're taking out one gene, but you're taking out 10 genes, of which 3 of them are related, that you would never see that type of scenario by sequencing even 100,000 exomes type of thing.
So you would say about 30% of the recurrent mutations, well, recurrent mutant genes overlap with the CNVs.
That's correct. Okay. And then there are some that we never see as CNV in anybody. And these are particularly very gene-dense areas where I'm sure there will be a CNV, but it's just going to be a smaller CNV below the radar, right? So it's basically not a space that's available to large events.
Thank you.
Yep.
Hi, Evan. Great talk. So I was thinking a lot about something that you mentioned with respect to the mothers being, and maybe buffering being there. And sort of a comment and maybe a question like what you think of this comment. What about the idea that mothers and fathers, or men and women, might be different in terms of how likely they are to be part of one of your pedigrees, which has 2 parents and a number of children, rather than it being buffering in terms of intellectual disability, it's buffering in terms of ascertainment, being part of that kind of a family.
So I should point out that those numbers are based only on families where both mothers and fathers have participated. So there's no data, it's been corrected. We didn't take things, for example, mothers are more likely to be in the clinic with their kid than a father, 5 to 1, right? That's always the way we see it. But in terms of essentially the transmission, we only looked at those where we actually had complete information from both the mother and the father.
Right, but what I'm saying is that there might be individuals out there who just don't have children and therefore don't become possibly part of your study. And there still is an ascertainment thing. I think it has to be considered in terms of the, buffering, of course, is also an interesting idea, and it's been discussed with respect to intellectual disability for years. But I think here you have another impact also just of the predilection to becoming part of a family like this.
I see. Yeah, that's a good point. I guess I would answer it in part this way, is that there are diseases where there's been no maternal bias, or I should say, no gender bias ever reported associated with the phenotype. And there have been studies, especially on immune-related genes, associated with Crohn's, lupus, and other types of things, where there's, in fact, the bias goes the other direction. So it would have to be something very specific about, I would say, neuropsychiatric, neurocognitive diseases, right, that would do that. If we could do grid sampling.
You wouldn't be far-fetched to hypothesize.
That's right, because it's
possible.
How people interact.
So you mean just more non-traditional quad trio type families that would be out there?
No, I'm not necessarily coming up with a way of fixing this. It's just something that you have to consider also as a possibility for the data that you observe.
Yeah. So the part that I really want to do, and this is a very difficult thing, and we have got it through IRB, but it's loaded from a kind of from a number of reasons. But what we want to do is we have certain genes now that we have risk, and they're almost always inherited. And what we want to do is we want to go back to the carrier parents, and we want to determine if they are truly asymptomatic or if they're in fact subclinical. We've done this with some of the CNVs, and we had a CNV years ago we published in Nature Genetics. It was not 16p11.2, it was 16p12.1. And it was one that we always saw transmitted. And we basically showed, based on large numbers of screens, that the odds ratios were through the roof. This was an important variant. But it was never seen in, except maybe one family, it was never seen as de novo. So we went back to a small subset of the families where there was a psychiatric evaluation had been done on both the mother and the father prior to determination of carrier status. Remember, we were ascertaining for kids with developmental disabilities, right? So it's pretty severe, and the parents did not have developmental disabilities. What was interesting about the carrier parent, in that study, 8 out of 9 of the carrier parents had a severe neuropsychiatric condition that ranged all the way from bipolar to major depression for 20 years. These are really hardcore neuropsychiatric deficits, and 1 out of 9 of the non-carrier parents. And at that point, our numbers were too small to consider gender at all in terms of the parents. It's a good point. But I think this will be solved in the context of the individual families. I really do. When we start going back and we can start to screen, maybe even biobanks, we were talking about that this morning here, right? If you could start to screen for these types of mutations and work your way back, and you could actually ascertain phenotypes, totally in a genotype first way, that would be, I think, a way to solve this.
Thanks.
That's good. Hey.
Hi. So you've discussed the fact that many of these gene-disrupting events, not only at SNV but also CNV level, they are basically grouping. So these genes are grouping in pathways or connected pathways, if you like.
Yeah.
Given the fact that many of these genes, they tend to have sometimes, even if they belong to the same pathways, they tend to have opposite effects. And many of the times they are actually quite differentially expressed in different, not only in the brain, but like the opposite effects in the brain versus gut.
Yep.
So we all would like to have, let's say, the therapeutics be applied at the pathway level. So how do you think that this is going to go? How do you think you would like to see this moving forward, given this heterogeneity, and also given the fact that many of these autism patients also have other comorbidities, right?
Yeah, yeah. Well, I guess the way I see it, but this may be a little bit too naive, I keep telling my postdocs this, I think the way to move forward with all of these genes, that once we've kind of planted the flag and we have the phenotypes and we want to explore this kind of opposing versus in wet tissues and what have you, is to start to really systematically do this. And I mean systematically do the mouse models, do the models in Drosophila, do the models in zebrafish, because I think there's a lot you can learn about that type of analysis that may in fact direct you. Because you realize when you're dealing with humans, even where, the strength of humans is there's 7 billion of us. But the problem is that in order to really address these types of questions, you really have to get into the tissues, you have to get into the stages of development. And in a sense, I guess the good news is because many of these genes are highly conserved, they're probably they're more apt to lend themselves to essentially studies in model organisms than, let's say, other genes that may have more recently evolved. So I encourage all of my postdocs that need, to develop a functional arm. I mean, I'm not a functional lab. I mean, that's not what I do. I'm a geneticist, and I'll always be until they take my money away or I die, right? But I know that this functional aspect is so important. And even for phase 1, phase 2 trials, you've got to have models. You've got to have test efficacy of drugs on those models. And I think that we should do this in a smarter way, because what I'm seeing, it's still happening. I mean, there's been some discussion about this, but what typically happens is 3 labs jump on the mouse at the same time. They start doing it, and the mouse, one of the 2 labs did it wrong. 2 of them don't have enough power. They do this analysis, 3 or 4 papers come out, everybody disagrees and agrees, and at some time down the line, it's like, oh, it's this. And I just think that this could be like an obvious next step. I mean, there are labs that do this, and they can do this in a high throughput way, just like the exome and the genome sequencing was done.
If you allow me one related question, because also combined with the therapeutical question I had before, but so if you think that many of these genes, especially, for example, the chromatin remodelers, are also associated, not on sporadic part, but with tumors, right? So how do you think that we have to think differently in terms of therapeutic, between autism versus, for example, colon cancer or things like that?
Yeah, it's a great question. Also a loaded one. I got in trouble one time for answering it, but I'll try not to get in trouble this time. So the question is, and maybe it's taking a little bit of different tact, is that many of the genes that we've implicated in terms of de novo and heterozygous are genes that are already known in the cancer field as actually being either drivers or very close to drivers of specific types of cancer. So for example, the CHD8 gene, that gene was associated with gastrointestinal cancer before we associated it with autism. At least I think that's right, or it's around the same time. And it's interesting, I don't know if any of you picked up on it, there was one family that was transmitted, the CHD8 loss of function, so it wasn't everyone. That, when we tried to go back and recontact that, we did recontact the family, the father had died at the age of 45, and he had died of colon cancer. And his wife swore up and down that he was autistic, but we didn't put that in the paper, because my wife might swear up and down that I'm autistic. But the important point here is, and it's one that you have to be very careful of, it may be that these kids, if we get enough of them and we follow them through time, may be at risk for GI-related cancers, because they already have the one hit in the Knudsen model, right? This is actually a really important consideration. But the flip side of that is these families have more than enough to worry about right now. And the thing that you want to do, ideally, is inform the clinicians to maybe do early assessments, right, on these kids to make sure they don't die of colon cancer at 25 or 35 or something to that effect. And with respect to the drugs, it's interesting because people are targeting drugs, and there are drugs that are efficacious. They work, right? They seem to improve. But what I always struggle with is in terms of the peak expression of the gene in development, it's always so bloody early, 7 to 8 to 10 weeks. How can you imagine any type of therapeutic at that stage, unless you can apply the therapeutic postnatally somehow? So yeah, I think, for people that are working on the synaptic plasticity stuff, which is postnatal, those are much easier. Sodium channels, calcium channels, they're all good, right? But when you're talking about a chromatin remodeler that affects, 400 genes minimum early in development, starting to tweak with that, it's got to be a recipe for disaster unless you're absolutely certain what you're doing. And that goes back to having the models.
Thank you.
One last question. I think Judy, go ahead.
Yes, wonderful talk. So my question is just a speculation about the genetic architecture between different types of diseases. So autism versus autoimmunity, we kind of have opposite problems. We had too many GWAS signals and not enough monogenic forms of disease, and maybe where the real value moving forward for functional was in this oligogenic space. And so in IBD, we're seeing, for example, that you have rare risk alleles, certainly not the odds ratios you're talking about, but they're co-segregating along pathways. So maybe the oligogenic model is the way forward.
Yeah, no, I'm a big believer in it. But I think that the problem that study section always throws at you is how do you, you basically, when you start to assume 2 or 3 genes, where's your power to prove? And that's the tricky business. And it may be, coming back to kind of the GWAS model, it may be that once we condition, so the other problem with a lot of these de novo events that look like they're as close to necessary and sufficient is this variability in expressivity, where some kids can be severely affected versus mildly affected. And I'm betting if you had enough patients, right, that you could look at, you might be able to do a GWAS conditioned on that same genetic variant and start to find the modifier loci. But you'd obviously need large numbers. Yeah, I mean, I had this discussion. It was started as an argument. It turned out to a total agreement. And it was basically, between me and David Altshuler a few years ago, about the relative importance of common versus rare. You remember that whole kerfuffle?
You've really changed a lot.
I've always been.
I heard you say this talk for a while, and so you haven't changed the direction, right?
Yeah,
I think
yeah.
Because it has a lot of it yields a lot, but you are now much more open to the fact that expressivity and other issues may in part be contributed to by the inherited, the less penetrant, and that may in fact ultimately be part of how we understand.
I would say the thing that influenced me the most was probably the schizophrenia
Right.
data on the very large numbers of samples.
Right.
I mean, those types of things are telling you that there's obviously a lot of value in these types of variants as well, but you have to get to the numbers. But it's funny, we reach the same conclusion.
Right.
I mean, I think the only big difference, but I mean, you may disagree with me, I think that the targets that we're identifying, even though they're only good for maybe 10, 15% of patients, because they are proteins, right, as opposed to non-coding elements right now, they are in principle actionable in terms of therapeutic targets. And to me, it's like getting to the families, right? So it's like, okay, we got a gene, and we have a gene that people have actually started to develop therapies for, right? Maybe for a different disease, but it's something where it's already been studied. Like, the story with DYRK1A, there's a lot of stuff on that, right? And for some of the sodium channels, there's already drugs that have been FDA approved, right? So there's knowing that your kid has that. I just think it's common sense to me that if you have such a heterogeneous disorder, and autism isn't as bad, let's say, as something like intellectual disability, which is even more genes, it matters if your child got the disease because of a mutation that results in microcephaly, or a mutation that results in an overgrowth associated, and you can't possibly imagine that if there was going to be a drug that's going to help them, that you would just apply it to all of those kids, because in some cases it would exacerbate, right? So I think to me it's you go where the money is, right? In this case for geneticists, it's where you're getting the strongest signal, right? And if you don't get signal here, then you got to go somewhere else, right? But the epiphany, if it's not really an epiphany moment, comes when you just finally accept the fact, well, of course all genetic variation is important. That's what human genetics has taught us for 60 years. Common, rare, inherited, de novo, CNV, SNP. The problem is that we tend to balkanize, as communities, because we see signal somewhere and we say that's the way that's where all the truth will lie, and in fact it's going to be kind of obviously it's integration, but
because I always think of humans, we are just the integration of all. We are more than just, but we are in part the integration of all of our DNA variation.
Yeah.
Some
I think
of it's new, some of it's inherited, and
And you got to start somewhere. Yeah. No, it's a good time. So for students and postdocs, I mean, these battles are really kind of becoming almost moot. And so, because in the old days, i.e. 5 years ago, you had to either raise the CNV flag or the point mutation or the Mendelian or the, there were all these different groups of people, but now it's really not the issue anymore. The question is, how do you integrate? And the other issue, which I still think is underestimated, is that we think we're good at it in terms of discovery variation, but I think we still suck at it. And I think that's a hard lesson, because when you've spent all that money and time and you pat yourself on the back for a 30x genome.
And because you've written in your justifications that this is going to be good enough to find X amount.
But if you're missing 80% of a specific class of variation, you cannot be happy with that, right? So if anybody who's an experimentalist, we need longer read technologies, longer than what's out there, even for PacBio and MinION. We need something that gives us 150 kilobase reads. And the day we stop aligning sequences to a reference genome to find variation is the day that all human geneticists win. Because when we assemble genomes and we create 2 genomes, haplotype 1 and haplotype 2, that's when I think we'll really have a good shot at solving the rest of the pie.
Well, thank you so much for a fabulous talk.